I would like to formally welcome everyone as they come into the room. And I also would like to formally welcome and say hello to Professor Sophia Lunt. She is professor at Michigan State University, assistant professor for biochemistry and molecular biology, as well as chemical engineering and material sciences, showing a big breadth of expertise. And she has gained this expertise starting with a PhD at Princeton University with Josh Rabinowitz from 2005 to 2010, then as a postdoctoral fellow at the MIT under Matthew van der Heijden really in cancer biology and related types of biological systems, where she then took those ideas over to Michigan State University and focused since then on understanding metabolic reprogramming in cancer, but also on the way she has a range of active grants like a breast cancer breakthrough award, where she's principal investigator, a next-gen grant for transformative cancer research, then also Michigan State University strategic partnership grants, MetaViver Early Career Investigator Award, even an NSF Career Award as a principal investigator, but also ranges of honors like the MANA Young Investigator Award from last year at the 2020 conference, and a range of others starting in 2002. So for the last 18 years, she's been collecting awards, and it's my great pleasure to welcome Professor Sophia Lunt and giving us a well-expected talk about deciphering metabolic reprogramming in cancer. Sophia, thanks for joining us.
Well, Oliver, thank you so much for that wonderful introduction and for the opportunity to present our work today. I really wish I could be in California in person right now. It's 18 degrees here in Michigan, covered in snow, and the wind chill is 2 degrees. So I hope I can come back in one of the future winters and enjoy the warm weather over there. So today I hope to tell you about the work we've been doing in my lab for the past 5 years. So I'll first give a brief introduction about cancer metabolism, and then I'll let you know what we've been up to. So we've actually known for a very long time that cancer cells have fundamentally different metabolism compared to normal cells. So as many of you know, almost 100 years ago now, a German scientist named Otto Warburg observed that unlike normal cells that take up our favorite sugar, glucose, and oxidize it to carbon dioxide using oxygen and our mitochondria, cancer cells take up a lot more glucose and they ferment it to lactate. And they do this even when they have plenty of oxygen available. So this process of taking up a lot of glucose and fermenting it to lactate, even when there's enough oxygen available, is known as the Warburg effect. This observation is very confusing from an energy or ATP point of view, because converting glucose to carbon dioxide is super efficient in terms of ATP production. And one molecule of glucose can generate up to 36 molecules of ATP. But if you ferment glucose, if you take glucose and ferment it to lactate, that will only generate 2 ATPs. So normal cells will only do this when they're forced to, right? Like when you're exercising and you're running out of breath, you're not able to deliver enough oxygen to your muscles, then your muscles are forced to ferment glucose to lactate. But cancer cells seem to be doing this all the time, and it doesn't make sense. Because it doesn't make much sense, Warburg concluded that cancer arises from defective mitochondria, that cancer cells are forced to ferment glucose into lactate because of impaired mitochondrial function. But it turns out that that wasn't quite right. So any of you who have done C13 glucose labeling experiments know that you get a lot of labeling and TCA cycle intermediates in the mitochondria, right? And it turns out cancer cells very much depend on their mitochondrial function. And even if they don't put a lot of glucose molecules into the mitochondria, they'll take other carbon sources, like glutamine is a favorite, and they'll send it into the mitochondria. And if you inhibit their mitochondrial function, or if you remove their mitochondria, Cancer cells really suffer, and in some cases, they cannot survive. So it seems that cancer cells actually choose to ferment glucose, even though it's energy inefficient. And to this day, we don't completely understand the Warburg effect. Well, before scientists could figure out the mystery of the Warburg effect, the field largely lost interest in metabolism for a while. And this may be partly due to the discovery of p53 in the 70s. Of course, p53 is known as the guardian of the genome, and more than half of human tumors contain a mutation or deletion in the p53 gene. So that was a big deal, a huge finding. And the Human Genome Project also turned people's attention to the genome and genomics, and metabolism was kind of forgotten about. But as we studied these genes, these oncogenes and tumor suppressors, it turns out that these genes directly impact metabolism. So for example, p53, MYC, RAS, PI3K, AKT, all of these help control glycolysis. SRABP upregulates lipid metabolism. MYC, mTOR, both affect glutamine metabolism as well as protein synthesis and nucleotide metabolism. So I think then researchers started becoming interested in metabolism again. In addition, we realized that we can take advantage of this Warburg effect, this addiction to sugar, to image tumors. So we can give patients radioactively labeled glucose, and organs that are highly glycolytic will import and concentrate them in that area, and then we're able to image those organs. So here you can see the image of a pancreas, which normally does not take up much glucose. But in this case, this patient has pancreatic cancer, and these cancer cells are taking up excess glucose, and it allows us to diagnose this patient with pancreatic cancer. So being able to take advantage of the Warburg effect to diagnose patients is great, but of course, we really want to take it a step further and take advantage of it for treatment. To further our interest in cancer metabolism, we realized that antimetabolites, which target metabolism, are already used in cancer treatments. So antimetabolites are chemicals that structurally mimic biologically active molecules. So it allows them to competitively bind to the active enzyme, and it doesn't allow the natural substrate to bind. So here on this slide on the left, we have the structure of folate, which is an essential metabolite. It's especially important for pregnant women, right? Where we have a lot of proliferating and dividing cells. And on the right, we have the structure of methotrexate. It's one of the first chemotherapeutic agents to ever be used. And you can see that it really mimics the structure of folate. Other antimetabolites that are used for cancer therapy are listed at the bottom. And they include pemetrexed, gemcitabine, 5-fluorouracil. The problem with these antimetabolites is that they do not specifically target altered metabolism in cancer. Instead, they target metabolism of all fast proliferating cells. So this is why we get harsh side effects, right, for cancer patients. There's a lot of gastrointestinal malfunction, baldness, nerve damage, and so on. So there's a lot of work to do, and we really have to understand a lot more so that we can improve our antimetabolites to specifically target metabolic pathways that are only upregulated in cancer cells or minimally upregulated in normal cells. So this combination of realizing that genes impact metabolism, that we can take advantage of the Warburg effect for PET imaging, and seeing the promise of antimetabolites have led to altered metabolism becoming an emerging hallmark of cancer. And in addition to this renewed interest in cancer metabolism, we now have very sophisticated instrument that Otto Warburg and his colleagues did not have, including mass spectrometry-based metabolomics. So my lab is interested in understanding alterations in cancer and coming up with better therapies by combining cancer models with the latest tools in molecular biology, including mass spectrometry, genetic engineering, and fluorescent agents. And I had the opportunity to pursue my research interests with some great trainees over the past 5 years. I had 3 postdocs, Xiao, Lei, and Ilana, 3 graduate students, Marty, Deanna, and Elliot, and 6 undergraduate students, Chua, Mayank, Leah, Toinu, Nate, and Joyce. So I'll be telling you about their work. So I'll take you through 4 projects that we've been working on: metastasis and metabolism, altered enzymes in cancer, cancer heterogeneity, and next-gen cancer imaging and therapy. So first, we'll start with the metastasis project. This work was spearheaded by an excellent postdoc in the lab, Dr. Xiaoting Teo, and he also mentored 2 undergrad students, Alita and Nate, and it was funded by Agilent and Metaviver and yielded 4 publications. So metastasis, of course, is the process by which cancer cells leave the primary tumor and spread to new sites all over the body. Metastasis accounts for almost 90% of cancer-associated deaths. And for breast cancer patients, for example, common sites of metastasis include the brain, the lung, the liver, and the bone. Now, for patients with early-stage breast cancer, the 5-year survival rate is almost 100%. But by stage 4, when cancer cells have spread to distant organs, the 5-year survival rate plummets to about 20%. And 30% of all early-stage breast cancer patients will eventually develop metastases, so it is important to come up with therapies for metastatic cancer. So to study what metabolic pathways might be important in metastasis, the first step was finding the right model. Just as there are many different strains of bacteria, there are many different strains of mice. So on the right, you can see a phylogenetic tree that shows some of the mouse strains that are commonly used. And some are closely related and genetically similar, and others are not. So if we study just one strain, we run into the possibility that our discoveries are specific to that strain only and not very relevant to metastasis in general. So to solve this problem, we teamed up with Kent Hunter from the National Cancer Institute, as he has been working on modeling the genetic diversity found in human breast cancer using lots of different mouse models. So what his lab did was take a highly metastatic breast cancer model called the polyamidal model and cross it in with lots of different backgrounds. And what you get are progeny that have very differing metastatic propensities. So they all develop mammary tumors, but their metastatic potentials are very different. So for example, if you cross the polyamidal T to a FBB background, you develop a certain number of metastases, and we'll call that a metastatic index of 1. And that's highlighted in this dark red rectangle. If you cross into the MOLF strain, then you get 5-fold less metastases compared to the FVB. So then we give them a metastatic index of 0.2. If you cross into the cast line, then you actually get twice as many metastases compared to FVB. So they have a metastatic index of almost 2. So then what we can do is map all of these back onto the phylogenetic tree and find matched pairs. So these are mice that are genetically very similar with extremely different metastatic potentials. So one matched pair is the BL10 and BL6. One is highly metastatic, one is less metastatic. Another matched pair is cast and MOLF. So my lab took tumors from these mice, extracted metabolites, and then analyzed them by mass spec. And we wanted to identify universal metabolic features of highly metastatic tumors. So we got a list of over 100 metabolites that were different, and we started looking at the ones that were statistically significantly different. And many of them had already been reported in the literature previously. So for example, NADH has already been reported to be present at high levels in highly metastatic tumors. This likely reflects high TCA cycle activity. Spreading to other parts of the body is a really stressful and energy intensive process. So they likely upregulate the TCA cycle. So it's good that our results are agreeing with previous findings. Another previous finding is that glutamine is present at low levels in highly metastatic tumors. Again, this likely reflects high TCA cycle activity because tumor cells really like to eat glutamine to fuel their mitochondrial activity and the TCA cycle. But one metabolite that had not been previously associated with metastasis is sialic acid. So we found that highly metastatic tumors have higher levels of sialic acid. If you're not familiar with this metabolite, sialic acid is a nucleotide sugar. So you take a nucleotide, which can make up DNA and RNA, and you take a sugar molecule like glucose and you stick them together. And they can modify cell surface proteins and lipids, and it can affect how sticky these cells are, how well they travel through the bloodstream, and whether or not they're recognized by the immune system. So we thought this was a really interesting metabolite to further investigate. Well, we noticed that in addition to having high levels of sialic acid, the highly metastatic tumors also have high levels of its downstream metabolite, CMP-sialic acid. So this is actually the activated form that can modify the cell surface proteins and lipids. But if we look at an upstream metabolite, UDP-N-acetylglucosamine, that's actually present at low levels. So it seems like there might be high glucose flux into this pathway. So we decided to look further into flux through this pathway. So we did steady isotope labeling studies, which are basically pulse chase experiments. And we did this using cell lines because it's a lot faster and cheaper to do these experiments. And what we did was give cells C13 labeled glucose, and glucose goes through a few steps in glycolysis, and then it can go into the sialic acid metabolic pathway. And so we wanted to measure how fast glucose carbons are getting incorporated into these metabolites in the sialic acid biosynthetic pathway. So we first confirmed that the cell lines we were using also have high levels of sialic acid and CMP-sialic acid. And in addition, they have much faster labeling of C13 into the sialic acid pathway. So they're putting C13 glucose molecules into their sialic acid much, much faster. So they have bigger pool sizes and their labeling is faster, which means indeed flux through this pathway is faster. So it seems like this biosynthetic pathway is important, but does it have anything to do with human patients? Well, to check this, we decided to look at expression of genes in this pathway and see if it correlates at all with patient survival rates. So we looked at 3 genes in the pathway, NANS, NAMP, and CMAS, which are all important for making sialic acid. And we asked, is there any correlation to patient survival? And what we found is that patients who have high expression of any one of these genes develop metastases sooner, and they also die faster. So it does seem to be clinically relevant. So at this point, we saw that highly metastatic tumors and cell lines have high levels of sialic acid. They have high glucose flux into this pathway. And patients that have high expression of genes in the biosynthetic pathway develop metastases sooner and they die faster. And next we wanted to ask, but is it really important for metastasis? And to do this, we decided to knock out the rate limiting gene in the pathway, so CMAS, and we decided to knock it out using CRISPR-Cas9 from 2 highly metastatic cell lines, the 4T1 cell line and the 6DT1 cell line. And so first we wanted to confirm that we have successful knockouts, and you can see that we're able to get 2 clones that have no CMAS expression in the 4T1 cell line, and also 2 clones in the 6DT1 cell line. But we wanted functional confirmation that they actually have a knockout of CMAS. And so we did this by using mass spec. So in wild type cells, they do have a functional CMAS. So they should be able to convert sialic acid to CMP-sialic acid. And that's exactly what we see. We see that in wild type cells, they have low levels of the reactant, sialic acid, and high levels of the product, CMP-sialic acid. Now, if we knock out CMAS, then we shouldn't be able to make CMP-sialic acid. And that's exactly what we see. When we knock out CMAS, we get an accumulation of the reactant, sialic acid, and we're unable to generate the product, CMP-sialic acid. So we have functional confirmation that we actually have knockouts. Now let's see what happens to metastasis. Well, first we wanted to check proliferation. So if you knock out CMAS, does it cause tumors to grow slower? And we checked this by doing in vitro studies as well as in vivo studies, and we saw that there's no difference in proliferation. The cell lines grow at the same rate when we do in vitro studies on plates, and the tumors, the primary tumors at the injection site, grow at the exact same rate. But if you look at the lungs of mice injected with these cells, we see a huge difference. So with the wild type cells, you can see that the lungs are completely covered in metastases. Whereas if you knock out CMAS in the sialic acid biosynthetic pathway, then the lungs are completely clean. We also do this with our second cell line, and we see the same thing. With wild type, you see lungs completely covered in metastases. You knock out CMAS and the lungs are clean. So we can quantitate this and see a statistically significant difference. So at this point, we show that sialic acid biosynthesis is elevated in highly metastatic tumors and cancer cells relative to their less metastatic counterparts. Well, given the important role of glycosylation in mediating cancer cell behavior, we reasoned that other nucleotide sugar metabolic pathways might also support metastasis. So we decided to look into a second pathway as well, called the UDP glucose pathway. So first we started with patient data. We wanted to see if there's any correlations, again, between gene expression levels and patient survival. And if we look at all breast cancer patients, we see that high expression of the very first gene, the UGP2 gene, correlates with decreased patient survival, but not the other genes. So you can see for the first gene, the red line is lower than the black line, and it is statistically significant. But the next two genes, UGDH and UXS1, the red line and the black line are just basically right on top of each other. So there's no difference. Well, we know that breast cancer is a very heterogeneous disease with lots of different subtypes. So we wanted to further stratify the data by different subtypes. So we started by stratifying by the 4 intrinsic molecular subtypes: luminal A, luminal B, HER2, and basal. And we saw a difference just in the basal subtype. So in the basal subtype, high expression of the first 2 genes, UGP2 and UGDH, means those patients do worse. So they have worse survival. But then if you have high expression of the last gene, UXS1, those patients actually do better. We also stratified by estrogen receptor status. So estrogen receptor positive breast cancer usually does better. And if you're ER negative, then usually you have worse prognosis. And when we look at those patients, we actually see the same pattern. So having high levels of the first 2 genes means it's worse survival, and having high levels of the last genes means you have better survival. So if you have high expression of the first 2 genes, that means you would generate a lot more of this metabolite that I highlighted in yellow, UDP glucuronate. But if you have low levels of the last gene, it means you consume less of that metabolite. So both cases, having high expression of the first 2 and low expression of the last, means that you're going to accumulate a lot of UDP glucuronate. So we wanted to investigate this further. What if we knock out UGDH so that they're unable to accumulate UDP glucuronate? So we did the same thing. We used CRISPR, we knocked out that gene, and we confirmed that those clones cannot make UDP glucuronate. Then we wanted to see how it affects cancer biology. So we found that if you knock out UGDH in this nucleotide sugar pathway, we don't see any difference in proliferation rates, again, similar to the sialic acid story. But if we look at migration, we start seeing differences. So we first performed a modified scratch assay. So that's when you scratch the plate and see whether or not cells can move around to kind of heal that wound that we made. And if you do that with the UGDH knockouts, they have a harder time closing the wound compared to the wild type cells. And when we quantitate it, we see a statistically significant difference. We also did a second test. We measured chemotactic migration using a Boyden chamber, or also known as a Transwell assay. And we found that when we do this, again, fewer cells are able to migrate through the membrane to migrate, basically. So it is affecting their migration. We then inject them into mice, and we see similar results as in knocking down the sialic acid pathway. So you can see in the wild type, they have a lot of metastases in the lung, but if we knock out UGDH, the lungs are very, very clean. So we did this with 2 different cell lines, and we quantitated that both by the area of lung that's covered with metastases, as well as the number of discrete metastases, separate metastases in the lung. And by both metrics, we get a huge reduction in metastasis. So we show that sialic acid and UDP glucuronate are upregulated in highly metastatic tumors, and gene expression of both pathways correlate with patient survival. And knocking out key genes in both pathways significantly decrease in vivo metastatic ability. So next, I'd like to tell you about our altered enzymes story. And for this, we wanted to know, how do altered metabolic enzymes support cancer cells? And it was done by 3 trainees at 3 different stages, postdoc Lei, a graduate student Elliott, and an undergraduate student Chua. And it was supported by an AACR Next Gen grant and the Mayo Clinic, and it resulted in 3 publications. So we already talked about how the Warburg effect is exhibited by cancer cells, right? Cancer cells really take up a lot of glucose and they ferment it to lactate. Well, we think this is mediated in part by an enzyme called Pyruvate Kinase, because many normal and non-proliferating cells will express this M1 isoform, or PKM1, whereas proliferating cells and almost all cancer cells will express the M2 isoform, PKM2. So we wanted to look into this further. Let me remind you what pyruvate kinase does first. So you might remember glycolysis, right? You start with glucose and you make pyruvate, which can be fermented to lactate or imported into the mitochondria. Well, pyruvate kinase catalyzes the last step in glycolysis. It converts phosphoenolpyruvate to pyruvate. So as I said, normal proliferating cells have the M2 isoform. And if you take normal proliferating cells that grow really well and force them to express the wrong isoform, PKM1, these normal cells will stop growing. If you do the exact same experiment in pancreatic cancer cells so that they express the wrong isoform, well, they can grow just fine. And if you knock down all pyruvate kinase, both M1 and M2, so that they have very, very little pyruvate kinase, they can still grow just fine. So if we're going to try to target pyruvate kinase for cancer therapy, we need to figure out what's going on. We actually know how to rescue the normal cells. If we give them nucleobases, it actually rescues this phenotype. But we need to figure out what the cancer cells are doing to get around this so that we can try to target it or use combination therapy. So first, we looked at the metabolic profiles of pyruvate kinase knockdown cells compared to wild type cells. So this is a heat map showing changes in metabolite levels following pyruvate kinase knockdown. And blue means metabolites decreased after knockdown, and yellow means those metabolites increased after knockdown. And the first thing you notice is that there are definitely some colors. So cells did notice that their pyruvate kinase is knocked down, their metabolism was affected, but they're somehow getting around it. So we focus just in the glycolytic intermediates. So if we zoom in and look at glycolytic intermediates, you see some expected results, which is that we see a huge accumulation of upstream glycolytic intermediates, phosphoglycerate and phosphoenolpyruvate. So we're blocking pyruvate kinase, so you would expect those to accumulate. But we were surprised that we actually have similar levels of downstream metabolites. Pyruvate, lactate, well, lactate is different, so that's expected, but alanine was same. So we wanted to see what was going on. It seemed like they were still generating quite a bit of pyruvate. So to investigate this further, we again performed isotope labeling studies with C13 glucose, and we were surprised to see that even with pyruvate kinase knockdown, downstream metabolites are getting labeled with glucose carbons. So pyruvate, lactate, and alanine are all getting labeled with glucose carbon, even with pyruvate kinase knockdown. So this means that cells are somehow making pyruvate without pyruvate kinase, and how are they doing that? How are they reprogramming their metabolism to do this? Well, if we go back to our pathway map, we know that there's actually several different ways that you can get around pyruvate kinase. So one way is to use gluconeogenic enzymes, PP carboxykinase, and pyruvate carboxylase. Another way is actually to use an enzyme in the sialic acid pathway. It's called N-acetylneuraminate pyruvate lyase, and that reaction will actually convert PP to pyruvate also. Yet another way is to take up the upstream glycolytic intermediate 3-phosphoglycerate and then take it through the serine biosynthesis pathway through PHDDH, PSAT, PSPH, and then convert serine to pyruvate. And then finally, you can catabolize some amino acids such as cysteine and tryptophan to generate pyruvate. So we started investigating all of these possibilities. So we went back to our heat map and we noticed that the most yellow metabolite is phosphoserine. And if we look at serine, that's also yellow. And both of these are in the serine biosynthesis pathway. So we do our C13 glucose labeling experiment. And if you look at the C13 labeling into serine, it's way upregulated in the knockdown cells. So they have high levels of serine and they also have high labeling. Again, that means high flux through that pathway. We also did a multi-tracer experiment and basically figured out that cysteine catabolism significantly contributes to pyruvate production. So we found that pancreatic cancer cells are able to proliferate with pyruvate kinase knockdown, and the way they're able to do this is through the serine biosynthetic pathway as well as cysteine degradation. So next I'd like to tell you about our cancer heterogeneity project. And this was really spearheaded by a wonderful graduate student in the lab, Marty Ogrodziski, and it was funded by the DoD Breakthrough Award and yielded 3 publications. So as I mentioned before, breast cancer has many different subtypes and people classify them many different ways. You can do it based on hormone receptors, based on gene expression, history, cell of origin. But no matter how you do it, we definitely know that different subtypes have different prognoses. And there's some subtypes with targeted treatment that's available and some subtypes that do not. So for example, many of you know that triple negative breast cancer usually has worse prognosis compared to ER positive breast cancer. So we wanted to know if different metabolic pathways are upregulated in different subtypes of breast cancer to try to use that information for personalized therapy. And so to begin to understand metabolic differences in subtypes, we used a MMTV-MIC model, which mimics the heterogeneity observed in human breast cancer. And so these mice all start with the same genetic mutation, but they develop tumors that look very different. And from these subtypes from this mouse model, we decided to focus on the papillary and EMT subtypes because other researchers had already shown that they have very, very different gene expression profiles. So we reasoned that they might also have very different metabolic profiles. So we took these tumors from mice, again, extracted metabolites, analyzed them by mass spec, and then here's another heat map showing the results. And you can see there's differences between the 2 subtypes, but the colors that really pop out the most are metabolites in the pentose phosphate pathway and the nucleotide pathway. So we decided to investigate that first, and we decided to look at gene expression profiles as well. And when we do that, we saw this pattern emerge. So one subtype, the papillary, has high expression of genes in de novo nucleotide biosynthesis. And the other subtype, the EMT, actually have high expression of genes in nucleotide salvage pathways. So we try to kind of put all of this information together. So here's a pathway map. You start with glucose here, and you take it through the pentose phosphate pathway. And if you like doing de novo biosynthesis, so you want to make all your nucleotides from scratch, from your favorite sugar, glucose, then you need these red genes, and then you can make all the nucleotides that you need. So the papillary subtype upregulates these red genes and they make their own nucleotides. Now, if you feel lazy and you don't feel like doing all that work, like the EMT subtype, well, what you can do is just take pre-made nucleobases from your environment, and then express all the purple genes that allow you to convert nucleobases into whatever nucleotide you need. So that's what EMT are doing. So at this point, we decided to try targeting some of these genes. So we decided to target one in de novo biosynthesis, PPAT, and one in salvage, APRT. So first, let's look at targeting de novo nucleotide biosynthesis, which is preferred by the papillary subtype. And as expected, if you target that pathway, the papillary tumors are not able to grow. We knock out that gene and we could not get a single tumor to grow in mice. So that is very effective for targeting papillary. Now you do the same thing in the other subtype. Remember, the other subtype doesn't care about de novo biosynthesis, so they really shouldn't care about this gene. And when we do that, as you can see, it actually does affect them a little bit. The tumors grow slightly slower, but it's not super effective. Next, we try targeting nucleotide salvage, which is preferred by the EMT subtype. And you can see that it is more effective than targeting the wrong pathway, the de novo pathway. It's not super effective, but it's a little more effective. We target that pathway in the other tumors that don't care about salvage. And now what we see is that the papillary tumors grow even faster than wild type. So this tells us that if you're planning on targeting nucleotide biosynthesis or salvage or nucleotide metabolism in tumors, well, you really want to be careful based on the metabolic profiles of the tumor subtype you're treating. So if you have a tumor that looks like papillary, you should definitely target de novo nucleotide biosynthesis. And if you have a tumor that looks like EMT, you're better off targeting nucleotide salvage. And if you accidentally mix it up, if you accidentally target nucleotide salvage in the papillary subtype, you're actually going to make things much, much worse for the patient because it can cause the tumors to grow even faster. But this data shows that targeting salvage actually isn't super great for EMT. So we looked at other metabolic pathways, and I won't take you through all the data, but basically we also saw differences in glutathione metabolism and the TCA cycle. And we found that targeting glutathione in biosynthesis is actually super effective in the EMT tumors. So you can see on the top left here, if you target glutathione biosynthesis, you get a 98% inhibition in proliferation, whereas if you target that same pathway in papillary, you get 50-some%. If you want to target the TCA cycle, it's a lot more effective in the EMT compared to papillary. But if you want to target papillary, like we talked about, targeting de novo nucleotide biosynthesis is the way to go. So we show that breast cancer subtypes use distinct pathways for their metabolic needs, and these unique vulnerabilities might be what we can exploit for personalized therapy. So finally, I'd like to tell you about our cancer imaging and therapy project, which is a little different from what I normally do in lab. This is in collaboration with my husband's lab. He's a chemical engineer and developed molecules for solar cells, and we wanted to see if we could actually apply them for cancer imaging and therapy. So this was led by a grad student, Diana, as well as a new postdoc, Ilana, and they mentored 2 undergraduate students, Mayank and Toinu, and it's supported by an NSF Career Award and an internal grant from the university. And it resulted in 1 patent and 2 publications. So we're interested in developing fluorescent agents for tumor imaging and therapy. And just to give you a little background, fluorescent agents work as photosensitizers that become activated with a specific wavelength of light to either kill cancer cells, detect tumors, or guide surgeons in image-guided removal of tumors. And they hold great promise as therapeutics because theoretically, the photosensitizer by itself is harmless, and the light by itself is harmless. But when you bring them together specifically where there's a tumor, then you can kill the cancer cells. Now in practice, the photosensitizers on the market today are not completely harmless. They actually have a lot of non-specific toxicity, and they cause damage or death to normal cells. And so patients that receive treatment from these available agents actually have to avoid visible light for weeks after treatment. Imaging is also limited because visible light only travels a few millimeters through skin. So tumors deeply embedded within the body cannot be detected or treated. So to address these current limitations, we decided to develop photosensitizers that are active in the near infrared wavelengths. So today's photosensitizers are active in the 400 to 700 nanometer range. So that's the visible light, that's the light that we can see. And the problem with these wavelengths is that if you look, we have all of these biological components in our body, melanin, water, hemoglobin, that also absorb those visible wavelengths. And that's why you have a lot of autofluorescence and not very much penetration. But if we go past 700 into the near infrared range, you can see that absorbance of those wavelengths by our biomolecules falls by orders of magnitude. So that gives us much better resolution, much greater penetration, and reduced autofluorescence. So we decided to work with those molecules, and our true innovation came in the way we were able to tune these molecules for their toxicity. So we can take a fluorescent agent, make it into an ion so that it needs a counter ion. And everybody thought that the counter ion that balances the charge on the fluorescent molecule did absolutely nothing. But what we found is that if you switch out the counter ion with certain ones, it makes them very toxic for effective therapy. Or if you use other counter ions, it makes them completely non-toxic, so we can use them for imaging. So we first tested this in human lung cancer cell lines, A549s. And here we're looking at cell viability as a function of, as you increase the concentration of the fluorescent agent. And if we use counter ions here on the left, you can see that even at very, very low concentrations, with or without light, all the cells die. So those are cytotoxic, meaning they're just toxic all the time with or without light, and we don't want to use those. So we move into the counter ions here in the center, and when we do that, and we don't turn any light on, you can see that we can use fairly high concentrations, and the cells are still viable. Their viability is not affected. But as soon as you turn the light on, you can see the cells are dying. So this is perfect for photodynamic therapy. We don't want other cells that are not illuminated to be hurt, but where we shine the light exactly where the tumor is, we want those cells to die. Now, if we move to these counter ions on the right, you can see that with or without light at very, very high concentrations, these remain non-toxic. So they would be ideal for imaging. Because if we want to figure out where the tumor is and we shine the light on the whole patient, we don't want to kill all the cells that we've illuminated, right? We just want to be able to find them without hurting the cells. And then once you find them, then you can bring in the phototoxic agent and kill the cells that you want to. So we wanted to know where our fluorescent agents were localizing in the cell, and to find out, we incubated the cells with our fluorescent agents, and we also incubated them with various markers. So rhodamine will go to the mitochondria, the hoistane goes into the nucleus. And so when we image all of them and superimpose the images, we saw that our agents actually go right into the mitochondria, but not the nucleus. We also wanted to understand why they were toxic in different ways. So we looked at what reactive oxygen species are being generated with our fluorescent salts. And we found that the cytotoxic salts, the ones that are toxic all the time, generate a lot of mitochondrial superoxide without light and with light. So they're just always generating a lot of superoxide, which is why they're always toxic. If we look at the phototoxic ones that are only toxic with light illumination, you see that when we don't turn the light on, we have very little mitochondrial superoxide being generated, but when you turn on the light, we get very high levels of mitochondrial superoxide being generated. And then if you look at the non-toxic ones, they generate very little mitochondrial superoxide and cytoplasmic ROS with or without light. So we wanted to see if our imagers actually do better than the imagers that are available. So we looked at our agent paired with a toxic combination and also a commercially available agent. So this is how it's formulated. It's CY7 and its counterpart is CL. It's pretty expensive at $1,200 per 100 milligrams, but you can see that there aren't that many cells, and the image is pretty dim because these are pretty toxic. So we switched out the counter ion to our non-toxic combination. And when we do that, we get much brighter images, and we see a lot more cells that are alive also. Next, we wanted to make sure that we can use this to image tumors in live mice. So we took a stereoscope and we outfitted it with a special near-infrared light source. And we bought a very expensive near-infrared detecting camera that can detect up to 1,700 nanometers. And then I can show you some of the images we've been getting. So here is a bright field image of a mouse. You're kind of looking at the abdomen. These are the hind legs on the bottom and the forelegs on top. And then this is a tumor that we injected in the 4th mammary fat pad. And we also nared the fur off of its belly because we found that the fur fluoresces a little bit. So when we turn on the near infrared imager after injecting the mice with our fluorescent agent, you can see that the tumor really glows. So the tumor selectively gets taken up. The fluorescent agents get selectively taken up by the tumors, and then the tumor lights up. And in this case, the mouse is still alive, so we can do it while the mouse is under anesthesia. But we did sack the mouse and take out the organs. And when we dissect out the organs, we see that we get a little bit of fluorescence in the liver and kidneys. This is because those organs are responsible for removing these agents from the body. We don't see fluorescence in any other organs, but by far the brightest fluorescence is detected in the tumors. We also imaged some lungs. So here you can look at the heart here and the lungs. And we were not able to see any metastases by eye when we check these lungs, but when we imaged them, we're able to see these tiny little dots. So we're excited that we might be able to use our technology to detect very, very early stage metastasis before they can be detected by other methods. So we're currently working on trying to figure this out. So we show that counter ion modulation can tune the toxicity of fluorophores, and phototoxic agents are ideal for photodynamic therapy, whereas non-toxic agents would be great for cancer imaging. So I'd like to thank everyone that contributed. I try to highlight individuals that contributed to each project and the funders. So these are all my students and postdocs, our collaborators, both at MSU and at the NCI. And thank you all so much for your attention. I'm happy to take questions.
Thank you so much, Professor Lunt. That was a fantastic talk. We learned a lot. Dr. Arpan Abania from our lab will now steer the discussion.
Great, thank you. Thank you, Dr. Lunt, for such a great talk. It's really exciting to see all of these research, projects related to breast cancer and how you implement metabolomics and other techniques like imaging to answer some key questions that your lab is focusing on. There are definitely some questions, and for those of you who are still with us, if you'd like to submit more questions, feel free to do so in the chat. So regarding your first project that you mentioned, the Metastasis project, you had shown some probability curves. There was a question about the cutoffs and what you use for high expression versus low expression for the enzyme transcripts.
Oh yeah, that's a good question. My postdoc would know a lot more about this. I think we have the details written in the papers that we published. So one is in Frontiers in Cancer Biology, I think, and the other one is in Cancer Letters. So we put all the details in that paper. I can't remember off the top of my head what cutoffs we used.
Great. Yeah, I mean, that is something we could follow up on reviewing the paper, of course. Alongside with that question, there was a question regarding the alteration of sialic acid and phosphoserine, and if that would also result in abnormal lipid metabolism in lipids like phospholipids.
Yeah, that's a good question. I would expect so, but we did not do any lipidomics. But if we looked into it, I bet there would be a lot of changes.
So just kind of going off of that, what normal platforms do you use for your studies?
You mean like which mass specs?
No, like chromatography, do you routinely use lipidomics, Hillic?
We usually use reverse phase, just pretty standard C18 reverse phase columns. We do use Hillic columns every once in a while. For nucleotides, we do use an ion pairing agent to try to help retain those nucleotides on the C18 column, because otherwise they just kind of come right off. But then it's a nightmare if you want to switch from negative mode to positive mode, because as soon as you switch, the ion pairing agent overwhelms everything.
Oh, wow. Yeah, I've never had experience with ion pairing, but have heard about the specific, technicalities of using that chromatography. There were some questions on your second project with the altered metabolic enzyme. So when you were specifically talking about making pyruvate without pyruvate kinase, how can glucose consumption and its related pathway in cancer cells, such as UDP glucose pathway, affect one sugar metabolism, or like other sugar metabolism, such as fructose? It's kind of a loaded question, I guess.
A lot of things there.
Yeah.
Yeah, I mean, I think so. We find that whenever we inhibit downstream enzymes, it does seem to upregulate upstream pathways. So we didn't specifically look at those pathways, but we think there's a lot more flux going through the nucleotide sugar pathway in addition to the serine pathway, probably. As far as how it affects folate metabolism, you know, folates I actually studied in graduate school, and so I love folates. But we're having a lot of trouble studying folates in mammalian cells because they're bound to proteins and it's hard to release them so that we can detect them. So I have no idea what's going on with folates, but I would love to know.
Thank you. I really loved the images in the Next Gen Cancer Imaging project that you're doing with your husband. Really great work. And there was a question around that project, but maybe it's a little general. They had asked, what makes the different breast cancer subtypes prefer different routes of synthesis? Do they have different transporters, for instance?
Yeah, why do they do that? I mean, I have no idea. Likely the different genetic alterations they accumulate over time might cause them to do that, or it could be the environment, like how close they are to the blood vessel or how hypoxic they are. It could be that. Yeah, as far as trying to figure out why they develop certain preferences, yeah, it's a good question. I'm sure people are trying to understand that. And as far as the imaging, wait, was there an imaging question also?
No, there are some imaging questions in the chat.
Oh, okay.
So there, one specifically is saying that while these imaging tracing agents are different in ROS generations, which I'm not really familiar with what ROS stands for, do they differ a lot in clearing rates from the body?
That's a good question. So ROS are reactive oxygen species, and that's what's generated by these fluorescent agents when they have light shined on them, or in some cases without light. Is there different clearance rates from the body? Probably. I'm sure there's different detoxification routes, but how fast they're cleared, I don't know the details of that.
Okay, thank you. As far as the imaging goes, is that something that would be costly for a patient, or what are the goals of, the end goal of that project specifically?
Yeah, I mean, the goal is to be able to image patients and treat patients with very, very, very little side effects. So our goal is to not hurt any of the normal cells and only hurt the cells where we're shining the light in the case of therapy. And in the case of imaging, to not hurt any cells at all, just to find them. So the end goal is, I mean, will it be expensive for the patients? Probably. I mean, hopefully not, because it'll be covered through health insurance. But the end goal is to be able to mass generate the key molecules that we can inject into patients And then the biggest cost will be the hospitals because they'll need big near-infrared images, right? They'll need cameras that can detect this. And just buying a little camera for the lab was very expensive. So I'm sure trying to outfit a hospital with a big whole body near-infrared imager will be very difficult, or maybe just have to image in sections, like small sections of the body. But hospitals already have extremely expensive equipment. So, you know, if it benefits the patients, I'm sure that's something that we can implement in the future. But that would be the goal.
Yeah, that's amazing. Yeah, just by seeing that picture of how all the cells lit up in purple was just, you know, mind blowing. Going back to ion pairing methods, there's a question in terms of what reagents were used for nucleotide liquid chromatography, I believe,
So
LC.
the ion pairing agent we used was tributylamine. I think that was it. Yeah. And then we used for our chromatography, again, this is in our papers. I think we used methanol. Oh no, it's a special formulation because now I remember making it all the time when I was a postdoc. We would have to dig it out of the papers, but I think we definitely have it in our cancer research paper.
Okay.
Yeah,
Great.
in the methods section.
And there was a question going back to the PK workarounds. Two questions here. What upregulated serine synthesis? ATF4, or I guess what are some upregulated serine synthesis? ATF4. I'm not really sure what ATF4 stands for. Maybe Oliver, you can help me on there. No? Or David, if you can explain what that is. And then the second question there was, did cysteine catabolism cause GSH stress? A lot of acronyms today.
So what upregulates serine biosynthesis? We don't know exactly what upregulates serine biosynthesis. You know, is it just that you're getting such an accumulation of 3-phosphoglycerate, and then the cell doesn't know what to do with it all, so then they start upregulating PHDDH, which is an enzyme that's also very hot in cancer metabolism right now. So what's the initiating factor that starts upregulating serine biosynthesis? I'm not sure. Metabolically, you would think, you know, you just have a ton of phosphoglycerate available, so you have to do something with it. It could be that. And does it affect GSH biology? You know, it probably does, but we have not looked into it.
Okay, great. Those were all the questions in the chat and that I had off the top of my head. Maybe Oliver, you have some questions?
There was also still the cysteine question. Cysteine catabolism causing glutathione stress. So that was also coming from David. Did cysteine catabolism cause glutathione stress?
Oh, I see. You know, it probably would. That would make sense, but we didn't look into it.
Okay. Yeah, I think these were great questions and multiple research strategies coming from those. I'm very glad that we were able to host you today. And I think it's a very promising career. Wish you best luck. And for our center, I'd say if you ever need help for collaboration, lipidomics or so, we'd be happy to do. Absolutely. We do it a lot. For the serine question and the 3-phosphoglycerate, we have seen it a lot before, that also methylations are much more prominent in cancer, epigenetic, that is to some extent governed by a methyltransferase that is a nicotinamide methyltransferase, which we published about, that really fosters then the serine glycine pathway that adds methyl groups. So that it seems to be a almost common feature in different cancers, and also involved a little bit in stem cell biology. So that might be governing to some extent, but really the root cause of that is not quite clear in terms of upregulation of activity of those enzymes, I think. The questions about sugars are interesting too, that were raised by one of my grad students here. We have seen those also pretty active in various cancers, all the way from hepatocellular carcinoma that we just published the other day, but also before in kidney cancers, we've seen that, including towards sugar alcohols. So where quite a bit of flux is driven from glucose actually to sugar alcohols. Even when you try to upregulate UDP glucose pathways, that's where a lot of spillover got to the sugar alcohols. Maybe as a reservoir, not quite sure. So it's looked more to me more like a bystander than an actor. So this was, I guess, my take on these two questions as they were coming around. Yeah, so are there further questions by people who are listening in? I have seen that a number of folks from the UC Davis Cancer Center and also from pharmacology were in. They haven't raised any questions yet, but I do know that they are very active in cancer metabolism as well, including as also like looking at specific cells with nanostructures. Anyone from those colleagues? That's not the case. Kind of, okay. So I'd like to thank again our guest speaker today, Professor Sophia Lunt from Michigan State University. Best of luck and hope to see you soon in a nicer environment than a Zoom meeting.
Well, thank you so much. I really appreciate it.
Thanks everyone for joining.